Hi, and welcome to our next webinar in our Plantae webinar series. My name is Jason Padilla, and I'm your technical host for today's webinar. Next slide. Next slide, please. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I would like to give a special thank you to all our ASPB members for attending today's webinar. Your ASPB membership dues help make this webinar possible. For any of you who have not yet joined ASPB, you can join today using the discount code %10 to receive a 10% discount on your registration. ASPB members get early access to these webinars, and you can learn more about ASPB and all the opportunities we provide at aspb.org. Next slide, please. This webinar is recorded, and we've had so many other great Plantae webinars that we've done previously. And so if you're looking for other exciting talks in plant science and various other topics, you could check out the Plantae YouTube channel, and you can find all the other talks we've previously done. And so next slide, please. As you know, the registration for Plant Biology Conference is already open. This upcoming Plant Biology is taking place in Milwaukee, Wisconsin on July 26th to the 30th. And registration is open now. We're currently have our early bird registration. And so if you registered pretty early, you'll get a pretty good discount. Plant biology is uniquely positioned to bring together members of the plant science community, where you're able to acquire new knowledge, network with fellow plant scientists, and learn from experts and learn about the different advances in plant science currently. And this is really going to be a great opportunity for all different career levels. And you can learn more about the Plant Biology Conference at plantbiology.aspb.org. Next slide, please. Before we get started, I'd like to go over a few housekeeping items to make sure that you get the most out of your attendance of today's webinar. If you are experiencing technical difficulties, please let us know about those using the Q&A box, or you can email me directly at jpadilla@aspb.org. If you have questions for our guest speakers, please add them in the Zoom Q&A box. Our moderator will be sharing them with our presenters during the Q&A, which will be happening after all the talks. If you are having difficulty connecting or need to leave the webinar early, as I shared, this webinar is recorded. And so you'll be able to access them on our Plantae YouTube channel after everything is processed. And so be sure to follow the Plantae YouTube channel so you can receive notification when the new recording is uploaded. And so without further ado, I am now going to turn it over to our Plantae Fellows who will facilitate today's session. And so with that, take it away, Plantae Fellows, and enjoy the webinar.
Thank you, Jason. Hello, everyone. I'm Yuan Yuan. I will moderate this webinar with other Plantae Fellows. So today's topic is climate extremes and plant science. As we all know, climate extremes are profoundly impacting ecosystems and agriculture, creating both challenges and opportunities for plant science. So this webinar will explore innovative strategies and cutting-edge research driving solutions for enhancing plant resilience, productivity, and environmental sustainability. Today, we are going to have 3 moderators and 4 amazing speakers. So they are Dr. Justin Whitehill, Dr. Li Yan, Dr. Gravo Zinta, Dr. Jesse Lasky. Next slide, please. The previous slide, thank you. No, the previous one, the outline one. Yeah, so in the outline for this webinar, we will have first the speaker introduction and the moderator introduce themselves, and then we have Q&A sessions. So all the attendees, please type your questions in the Q&A box. And last, we will have concluding remarks. Next slide, please. So hello everyone again. I'm the first moderator. I'm Yuan Yuan. I'm a professor at Fujian Agriculture and Forestry University. My research group is interested on engineering plant metabolomic pathway to produce valuable compounds for human and plant health, utilizing cannabis as a model substance. So my team explores a wide range of techniques from biochemistry through molecular biology to multi-omics analysis, coupled with deep learning-based modeling. Next slide, please. So the first speaker, Dr. Li Yan, who is a research associate in the plant science at the University of Western Australia. Dr. Yan's research focuses on elucidating plant-soil microbe interactions, particularly plant response to diverse environmental conditions, crop productivity, invasion success, and biodiversity dynamics. So Dr. Yan, the stage is yours.
Thank you, Dr. Yuan, and let's just share my screen. Oh. It's in processing, so in a few seconds. Can you see my screen now?
Yes.
Yes, we can.
Yes, perfect. Thank you. Hi, everyone. Thank you, Dr. Liu Yuan, for inviting me for this good opportunity to share some interesting work with all of you. I'm Li Yan. So today I'm going to introduce the plant diversity in impoverished Australia, especially the role of phosphorus. So let's start from here. Phosphorus, we know that phosphorus is very important almost for all the physiological process. And at the same time, phosphorus is non-renewable resource. And in most of the ecosystems, it's the first or the second most commonly limiting nutrients for plants. And it's non-renewable, which means the more we use for today, the less we can use for the future. And at the same time, this figure can show you globally there are very large scale phosphorus limitation in natural terrestrial ecosystems. And what you can, this kind of a deep blue color means these land areas, they face very serious phosphorus limitation, especially here in the corner, the green circle can show you the location of Australia. It's very blue, it's very peat limited. And the soil total P here, just 20% of the global average. And but at the same time, from this figure, we can know there are totally 36 global biodiversity hotspots in the world, and we have 2 in Australia. So that's amazing. Especially, for example, in southwestern Australia, there are more than 10,000 vascular plant species here, and 6% of them are endemic. And so we will have a question. You can see this this kind of typical landscape, land view of Australia. They are beautiful, and also at the same time, the very dry, very challenging land. We want to know how plant species can adapt this kind of poor nutrients here. As many studies have shown that some plants, they are very special here. They can grow very well with specialized roots. You can see like this kind of brush-like roots from Proteaceae. And another figure can show you the scallop-like roots. They are from Cyperaceae. Most of these species, they are non-mycorrhizal. And with this very short lifespan, specialized roots, they can release lots of carboxylates to mobilize soil nutrients, especially phosphorus. And here, this can show you how root carboxylate works. Actually, there are lots of phosphorus in soil, but they are not available. Only form available phosphorus in soil is phosphatate, but it's very low concentration. Most of the phosphorus, they bond with other elements like iron and manganese or calcium. So they are not available for plants to directly take up and use. But with carboxylate, it can mobilize the phosphorus be bound with other elements and make it more available for plants. At the same time, other elements like ions, manganese, zinc, and copper be available as well. So this is how root carboxylate works. But not all species here can form this kind of amazing specialized roots like class roots or dorsaiform roots. Especially, for example, we have 800 eucalyptus here in Australia, and most of the non-mycorrhizal species, you may say they may rely on this kind of fungi partners, but I can tell you it's very hard. As here is so poor and impoverished with phosphorus, so the fungi partners, they can't doing very well. So we have a question: does this species they more rely on themselves for soil nutrients? We don't know that. So you can do carboxylate measurement, but it will be very hard in the field, especially we want to do hundreds of Plantae eucalyptus species. You can take soils, but it's time consuming. So it's really a challenge, but we can use leaf manganese concentration as a proxy. And the same figure. Let's see how it works. As we know that carboxylate can mobilize phosphorus at the same time with many other metal elements, so including manganese, but different from other elements. Manganese cannot be well controlled for plant to take up, so which means it will accumulate in mature leaves, which means it can be higher and higher concentration in plant leaves. And it's been well proved in many publications. So if we see some plants with very high leaf manganese concentration, it's a big chance it can release carboxylates to soil. And we both know that the soil conditions changes among different locations. So different soil pH, different manganese concentration in the soil, which means there is a big variation. So if you want to use leaf manganese concentration in mature leaves as a proxy for carboxylate, you will need to use positive and negative reference as well. So here in Australia, we normally use Proteaceae species. They can form class roots. And also we know that release lots of carboxylates from plant nutrients. We use them for positive reference. And in the right, we found some plants like a grass tree. These grow very slow and they can't release carboxylate. They don't have high leaf manganese concentration. We use this kind of species as a negative reference. So we have the first question. Why are some tall eucalyptus forestries in high biodiversity Australia cannot exhibit high plant diversity? So we sampled in both Western Australia and Eastern Australia, and we want to know how this kind of a root carboxylates common or not beyond Proteaceae or Cyperaceae. So in the past 3 years, we sampled more than 200 native species with 1,500 plant samples. And here, I would like to show you part of the results. The this is the leaf manganese concentration, and the yellow box plot can show you positive reference. The green shows you target species, and the gray box plot show you negative reference. We found different patterns. The very left, you can show this with the leaf manganese concentration is very similar with negative reference, which means they can't release carboxylate. And in the middle, it's significantly higher than the negative reference. And in the right, lots of species, they show even higher leaf manganese concentration compared to the positive reference. Those species can form specialized cluster roots. So here we found it's very common. I can see it's the first time we found it's very common, not just for Proteaceae species, but many other Myrtaceae species. They have high leaf manganese concentration, and probably they release lots of carboxylate. We don't know that until we picked 10 eucalyptus species with different leaf manganese concentrations. We do that in the glasshouse with different pre-treatment. And yes, we found some species, like in the bottom green circle, they don't respond to P, and they can't release carboxylate. Not a significant amount, but many other species, they do release lots of carboxylate to mobilize P, but not all species, they respond to phosphorus. So maybe carbon, we are still figuring out that. And we know for phosphorus strategies, one is getting as much as you can when you can get it, like what specialized roots are doing. And the second one is making the most use of what you have got. So when you have very limited resource, you better think about how to well use that. And here again, we pick Jurong Bay as a simple site. Here, from the very close to the ocean side and a little bit further to the ocean, we can find different soil stage. The younger soil stage, like stage 2, the soil pH is relatively high, but the older stage, very, very low phosphorus. So we found different species, they have different distribution here, like Fabaceae, Proteaceae, and Myrtaceae. And the result is Myrtaceae. They can very flexibly allocate and distribute the P from one fraction to another fraction. But for Fabaceae, it's always have very high requirement for phosphorus and nitrogen. So this species with NF, which means we can't find this species in very poor soil stage. So it can't do well compared to other species. Other species like Proteaceae and Myrtaceae, they can flexibly reallocate phosphorus chemical fractions, but some other species can't. That's why we find it's very important for species. Some species, they have the capacity to just adapt different kind of soil conditions. So this is the take-home message. Now we know leaf manganese is a very good proxy, and root carboxylate can provide significant advantages to either mycorrhizal or non-mycorrhizal species. And we have results as including invasive plants. There's another story. And but however, for eucalyptus, not all plants do that for phosphorus. There's another story. And the third one is the capacity to efficiently reallocate leaf phosphorus to key chemical fractions improve the adaptation for some species. The fourth, now we can see phosphorus oxidation and utilization strategies play very important roles. And that's my acknowledgement. Thanks for my supervisor, Hans Lambers, and my wife, Dan Tang, and all other colleagues contribute to these fantastic studies. And as editors, I would like to introduce these two journals for your consideration to submit your fantastic manuscript. One is plant soil, and another one is plant environment interactions. And also I have an academic channel. Welcome to introduce your papers, your lab positions, or any other things you want to share. And thank you. Welcome to the WA. Thank you, everyone.
Thank you so much, Dr. Ian. Great talk. Now we are moving to the second speaker. Next slide, please. Next slide, please. Our moderator. Okay, next slide, please. Right, so now we have our second speaker, Dr. Justin Whitehill. Dr. Whitehill is an assistant professor in the Department of Forestry and Environmental Resources at North Carolina State University, NCSU. He leads the Christmas tree genetic program at NCSU and co-directs the Forestry Biotechnology Group. He and his team are working to develop pest and pathogen resilience freezer firm, the Christmas trees, through genetic and genomic approaches. The ultimate goal is to accelerate genetic improvement efforts of conifers to meet the cascading challenges caused by a changing climate. Let's welcome Dr. Whitehill.
All right, thank you for the introduction and thanks for the opportunity to speak here today with everyone. You're able to see my presentation, okay?
Yes, we can. Thank you, Dr. Whitehill.
Yeah, thank you. All right, well, so I'll just give my talk today is geared more around an overview of our research program and the work that we're doing with conifer trees, which can really apply to any of the various conifers and the most economically important group of conifers and the Pinaceae around the world, which include pine, spruce, and in the world of Christmas trees is where I work, and the other group there that we work with are the true firs, or the genus Abies. So I'd like to put this in context. I know we're talking to folks all around the world, but Christmas trees are a big thing in the United States. They originated from Europe, but in Germany, and they are associated with, can be religious holidays, but also are becoming more and more secularized around the United States. Canada and Denmark are two of the other countries that have the largest Christmas tree industries around the world. But the idea of bringing green foliage, that evergreen materials in to one's homes during the holidays, during celebrations, is something that dates back to the ancient Egyptians over 8,000 years ago. So it's a tradition that's had a lot of various incarnations over time. And I always like to put this in context with this quote that the joy associated with the Christmas holiday evergreen ignites an appreciation for the beauty of trees, which is a long step towards the will and to plant and both care for them as well. So I like this quote because Christmas trees are often a point of entry for people in areas that participate in these celebrations. It's a way for them to encounter a tree. I know young children often will look at the trees, interact with them, think about the aroma, think about the sticky sap, and really marvel at kind of the beauty that they have. And it's a place where families sort of gathered throughout the cold winter months and celebrate. So it's a really nice tradition, and trees are at the center point of that. And so that's where our job comes in, is to really work with these species of trees, which are facing a lot of challenges around the United States and around the world, and conifers in general. But the most popular species or genus of Christmas trees in the United States are the Abies, or the true fir. They account for about 66% of the industry. You could also add that 7% in, because that includes other kind of miscellaneous fir species from around the world, or around the United States, so it's over 70%. The industry is valued somewhere around $2.5 billion in the United States. And in North Carolina, the value is estimated between 250 to 400 million, which is somewhat of a conservative estimate, as it leaves out some of the other associated industries that are a part of this Christmas tree work. But the biggest species by far that accounts for most of the trees that are sold in the United States are the Fraser fir. And the Fraser fir is a species that's native to North Carolina, but it faces some significant challenges. A little bit about this species, and I'm sure not many of you are familiar with it, but it's native to the state of North Carolina. It has populations that sort of spill over into Virginia and Tennessee. So this is a map, a kind of shrunken map of the United States on the east coast of the country, towards the southern half, but the state of North Carolina you can see here. And along the border here, we have the Appalachian Mountains. This is also called the Blue Ridge Mountains in this part of the world, or the Great Smokies, depending on where you are within that range. But basically, the Fraser fir is sort of a remnant of the last ice age. As the temperature started to increase over the last ice age, trees sort of began, conifers in particular, began to retreat north. And so we get the boreal forests in Canada now that have all kinds of conifers that dominate those landscapes in the more colder parts of the continent. But in some of the more southern range, as the continents sort of retreated, or sorry, the ice, the glaciers retreated, conifers began to sort of also retreat, but in some spots they were sort of geographically isolated towards the highest mountaintops. And those highest regions of the highest mountaintops in the Appalachian Mountains actually were very habitable for conifers. And so they formed some really unique ecosystems at the very tops of the tallest mountains and in the mountain ranges of Western North Carolina. And so we only have about 6 naturally occurring species or populations of the Fraser fir, but it's a species that's seen a lot of challenges, especially as climate change is increasing. We're getting a lot of new challenges that the species is having to face, and we're really worried about the longevity of the species. Of course, as global temperatures change, that unique ecological niche may disappear. So, and I would also mention that the species is endangered in the wild, not from the Christmas tree industry, but because of invasive pests that were introduced in the 1950s. Only about 10,000 trees actually exist in the wild. So the Christmas tree industry plants millions upon millions of trees every year, and the work we're doing is actually helping to conserve the species. But it's sort of known as the perfect Christmas tree, or the Cadillac of Christmas trees, which are very kind of regional specific terms, but the species itself has what we call kind of elite traits. It's very soft and nice to touch, which is good for decorating. When the tree is cut or harvested, the needles do not fall off, which is a really big complaint that most people have. Bringing a live tree into your home, you don't want the needles falling off. The aroma is very nice. It's sort of an aroma that's become associated with the holidays, and it's the #1 reason that consumers purchase a live tree, is because that aroma will permeate through their house for weeks on end during the cold winter months. And the branches are strong, which holds for decorations. But there are some major issues that it's facing. And one of the big things that we say when talking about this Fraser fir species and species within the genus Abies in general, the true firs, is that they're really global indicators of climate change, and they're sort of the canary in the coal mine species. So because Fraser fir has such a unique ecological niche at the tops of these tallest mountains in Western North Carolina, it's estimated that models have predicted an increase of 3 degrees centigrade in those climates. If that occurs, expected to perhaps occur sometime in 2050 or before, we could see these kind of unique ecosystems of Fraser fir and red spruce disappear in those areas. And so it's very important to us, if we want to conserve and keep this species around and help supporting the rural economies in the state of North Carolina and around the United States, that we figure out how to keep this species sort of going. And so in addition to climate change, there are all kinds of challenges that the species is facing. I won't go into all of these, but one of the biggest ones is the Phytophthora root rot pathogen. It's a disease, oomycete pathogen from Asia, that was introduced or hypothesized to be from Asia or Papua New Guinea area. And it spread throughout the United States in the 1950s, 60s, 30s, all over. And unfortunately, Fraser fir is extremely susceptible to this pathogen. And so as climate is changing, the environment is changing, and the pathogen is getting more and more favorable conditions to thrive. And the areas where the trees used to thrive are now very stressful for the trees, which makes them more and more susceptible to disease. And also these other challenges like insect pests, drought issues, as well as disease pressure from other pathogens, as well as even deer browse. Less food for deer in the winter means that the trees are actually a food source for browsing mammals. I like to put this in context, though. Conifers are being threatened all over the world. I spent 10 years working at the University of British Columbia in Canada, working as a coastal forest health research scientist as well with the British Columbia Ministry of Forests. And lodgepole pine that blankets the western part of British Columbia, as climate has increased over the last 20 years or so, the trees and conifer trees in that area have become more and more stressed, which has made them more susceptible to what's called the mountain pine beetle, a very voracious bark beetle that has killed hundreds and thousands of millions of trees on the landscape. It's really created a very bad situation in that part of the world. But this is happening all over the world for various conifer species and various continents. And when you have a lot of trees that are diseased or stressed on the landscape, that leads to a lot more forest fires, which is what we're seeing all over the world. So this is happening all over the place with conifers in particular, which are more adapted to kind of cooler, wetter climates. Our program in particular works on Fraser fir, but we also study some other species that have some natural climate resilience, like Turkish and Trojan fir, that can handle higher pressures of diseases and pests as well as climate. They don't have exactly the right traits, but they do have some genetic information that we're looking to understand. And then we also have a species from Japan that is 100% resistant to that pathogen that we is really the biggest challenge we're facing in our industry. So we're looking at all these different species to try to understand the underlying genetic mechanisms that are protecting them from these major challenges. Our lab really survives at the intersection of research as well as getting that research to our growers. We do that through our extension program. So really our research is built upon our interactions with the NC Christmas tree industry and our farmers that are out in the field. Our program itself has been around since 1980. I took over in August of 2020, so 40 years after the program began. I'm really the second, almost third person to take over this breeding program. But the idea is that we've been developing trees that have enhanced resilience to pest pathogens, higher dollar values, faster growth. And we're trying to get those out to our growers, and we do that through establishment of seed orchards, getting seedlings out that then we get to our growers who plant these enhanced and more resilient trees. So our program is just getting to the second generation of genetic improvement, comparing that to things like corn, which are on thousands and thousands of generations of genetic improvement. We have a long ways to catch up, but part of that is due to the slow timeframes of conifer tree breeding. Two ways that we develop new genetics in conifers are through gradual population improvement, or a classical quantitative genetics approach, or integrating genomics technologies, or through immediate functional genomics strategies. And this is really the only way that we're able to develop new resistance in our Fraser fir and particular species, as we've screened all the germplasm we have for this species that exists, and these trees are have no natural resistance. So that's why we're looking at other species to try to figure out how to integrate that resistance into those species. So developing resistance in trees and conifers in particular is very challenging. There's a lot of different ways to do it. In different examples, I'm just briefly talking about here, using hybridization like the American chestnut program with an Asian tree and a North American tree, identifying resistance within a population as they've done in British Columbia and Sitka spruce, introducing genes that can provide resistance from other species, which has been done with the American chestnut again, or using CRISPR to identify genes and single nucleotides that can be modified. So these are basically our options for developing resistance in trees, and we're really in the process of exploring all of those different approaches. Again, just to put this in context, Christmas trees were only through one generation of genetic improvement. Our most advanced breeding program for a conifer is loblolly pine, which is in its fourth, going on fifth generation, compared to corn, which is in the thousands of generations. So it's a very slow process, and we're only at the beginning here. I just want to briefly mention that we recently received funding from the Specialty Crops Research Initiative, USDA NIFA, to help accelerate genetic improvement in fir. It's called the Gift Seed Project. $7.5 million that was is aimed to engage growers around the world, really, with the US being the lead, Canada and Denmark, 6 universities, different companies, government agencies in various countries. And the goal of this is to work with our growers side by side to accelerate genetic improvement and provide guidance through genetics to these growers using advanced genomic and molecular tools. Just a brief shout out to some of our collaborators and colleagues on the project, these 6 different universities. We're actually meeting for the first time in person at the end of this week to really get the project rolling. Just a breakdown of the various species, but the big species we work on are Fraser fir on the East Coast. I should mention the next most popular Christmas tree species in the United States is the noble fir, with about 2 or 3 million, 4 million trees produced within Oregon, Washington state, and then Canada produces balsam fir. So those are really the 3 big species that we're looking at here. The industry itself around the United States consists of unknown seed sources to very well-known tested materials, but that challenge of not knowing where your germplasm comes from is really the basis of our genetic research that we're trying to do with this new project, is to try to develop genotyping tools to help understand the genetic relatedness amongst our trees. I will give a shout out for us that we are hiring. So we will have several postdocs, PhD student, master's positions, as well as technician positions. If you're interested, please contact me and pay attention to the NC State Job Board for more information. But ultimately, this work is all facilitated by the growers in the state of North Carolina and around the United States, and educating our growers about the importance of the work that we're doing in science and climate change and sustainability is something we really are striving to get out there. I think as we advance through time, as climate continues to change, these species and industries are going to face more and more challenges. And we're seeing that already in conifers. And the reason I'm so concerned is because they take so long to already develop new genetics that we need to be planning for 50 years in the future, and we're unsure what's going to be happening in the next few years. So with that, I'd like to thank my lab, my collaborators, and our research funding. If you're interested, please follow me on any of these platforms. And yeah, that's the end of my talk. So thanks again.
Thank you so much, Dr. Whitehill. Such a very interesting story of Christmas tree. So we are moving to the 3rd speaker and the 2nd moderator, Dr. Hao Chen.
Hello, everyone. My name is Hao Chen. I'm the assistant professor of the Auburn universities. And just as the Dr. Justin mentions, the Christmas trees is an important species in the North Carolina. I was studying in the NC State for 12 years. And I do not work in the Christmas tree, but I worked in the other important trees, such as the poplars and the pine. And my focus is for the wood formations, which provides the timbers and the pump to the customers. And currently, we focused on the drought resilience and how it affects the wood formations. And now, let me introduce for the Dr. Laskis. And could you move to the next slide? Thank you. Yeah, Dr. Laskis is an associate professor at the Penn State, and now he is also the director of the PAC's herbariums. And after the job as a faculties, Dr. Laskis focused on how plant adapt to the environment, and using Arabidopsis and sorghums as a model species. And previous to Dr. Laskis in the Penn State, he was studying the Columbia universities and UT Austin. Both universities are very prestigious. So now we placed looking forward to hear the Dr. Laskis, the excellent presentations. Yeah.
Okay, let me just bring up my slides here. Okay, how's that look? Good? Okay, thanks everybody. I am going to talk about Arabidopsis and genotype environment associations, which many people are starting to use for trying to understand the molecular basis of environmental adaptation. Am I still on the first slide or to go to the second slide? You seeing my second slide?
First
Yeah,
slides
second.
now.
First slide. Okay, something's wrong. This is my screen sharing is paused.
It looks good in our end. We can see your first.
Yeah, I had moved on to the second slide. If I It's not letting me advance. Sorry. You still see the first slide
This looks like your second slide.
or second? Yeah. I don't know why it's doing that.
If you want to keep it this way, I think maybe where your cursor is probably clicking, it's not on the right spot. But if you want to just advance from here, I think it's fine.
Yeah, I mean, I can advance. Okay.
Yeah, it's working now. I think it's where you were clicking.
Okay, so something.
It gets finicky, so just yeah. But right now it's advancing. So.
Sorry, that's the wrong one. Here we go. Okay, thank you. Okay, so a lot of people are interested in improving plants for future environments. We have lots of impacts that are probably going to occur for lots of different plants. This is a problem that a lot of us are facing. So one of the potential sources for improving plants could be some of the alleles and traits that are found in local populations of whatever plant species you work on. These different local populations may be adapted to different local environments. And because of that, though they may carry some alleles and traits that make them good at performing in those environments. One example of plants that have different local populations and different environments are crops, where you have these traditional local varieties all around the world for some of these widely cultivated crops. We do a fair amount of work on sorghum in this context, but I'm not going to talk about that a lot today. Really, I'm going to focus more on Arabidopsis in a moment. Now, an emerging approach to trying to identify candidate genes that are involved in local adaptation is to conduct genotype environment associations. So just a brief background on what these are. This is a pattern that people look for where you have an allele that is common in one environment but less common in a different environment. In this case, I'm showing you two different environments, A and B, and each of the lines there represents the genome of one individual. The black diamond indicates a mutation that is adapted to the blue environment, and it's only found on the individuals that come from that environment, which is a signature of local adaptation. So what people will do is look for this type of pattern all across the genome. One term that you can use for this is a genome-wide association study for environment, or an environmental GWAS. We had a review about a year and a half ago in the Plant Cell that was on this topic. So if you're interested more, you can go look there. Now, the reason why people are interested in these approaches is that they only require genotype and environment data, which are fairly easy to generate nowadays. You don't need to do lots of experiments. You don't need to do any experiments, actually. Now, this is also one of the weaknesses of these approaches, but there are some low barriers to entry for conducting such studies, which is one of the reasons people have been conducting many of them. Now, another potential promise of such studies is that we may be able to link together the specific environmental gradients with specific genes that are important for local adaptation. So one might be able to conduct these studies comparing a couple of different environmental gradients. In this case, I've got A versus B, which sort of might be temperature, and then we have X versus Y, which might represent soil type, let's say. And you can scan the genome looking for these associations, or if you have a candidate gene that already has natural variation, you can scan different environments and ask which environment is this natural variation associated with. And that might be the agent that drives changing selection across your landscape, maintaining that natural variation. This is part of the promise of the approach. However, one of the things that we wanted to, a problem we wanted to address with this review I mentioned is that many of the assumptions underlying genotype environment associations are often overlooked. So the favorite hypotheses that I highlight here are important, and they're often assumptions that are implicit in papers that use these approaches. So firstly, one of the first favorite hypothesis I have here is that individuals inhabit different environmental conditions. Of course, you need to have different environments to have any local adaptation to environment between your populations. Next, the populations have to be locally adapted. If the populations aren't locally adapted, that is, the home genotypes don't have home genotype advantage, then you're not going to find any environmental adaptations in those local populations to their specific environments. And I'm just showing you a cartoon of that where the home genotypes have higher fitness based on their size in their home environment. Another favorite hypothesis is that the different environmental dimensions are not confounded. So in this case, soil and temperature are not correlated with each other. Now, of course, there are often many confounders among many environmental variables are confounded in nature often. To a certain degree, this can sometimes be very hard to disentangle with observational studies, but study design can help that. Another important favorite hypothesis for this type of approach is that local adaptation is oligogenic, which means that there are a few genes underlying local adaptation having large effect. These can be detected with association studies, whereas if local adaptation is polygenic, then the importance of any individual mutation in local adaptation will be low, and the potential utility of any individual identified natural variant in a breeding program, let's say, is likely low. Another important favorite hypothesis is that the local adaptations, the black mutations here, are not confounded with population structure or patterns of relatedness, which drive the distribution of neutral mutations in space. However, there's often limited gene flow and slow turnover environments through space. So you often have neutral mutations being correlated to adaptive mutations. That makes it hard to identify which mutations are actually locally adaptive. This happens often in nature, but study design again can help. And then another favorite hypothesis for environmental GWAS, but also for regular phenotypic GWAS, is that we have genetic homogeneity. That is, a single allele in many individuals contributes or causes the trait variation. When you have heterogeneity or different alleles, different loci causing the trait variation, you have all the causal variants being more rare, which is harder to study with an association study. Okay, so just after having gone through some of that background, I want to give you a brief example of applying genotype environment associations in Arabidopsis. So we work a lot on Arabidopsis natural variation. In the upper right is an earlier diversity panel from which we had published some papers starting when I was a student, going into the beginning years of my faculty job at Penn State. I was using the data that came from the 250,000 SNP chip for about 1,000 Arabidopsis ecotypes from the native range. Since then, this has sort of been supplanted by the 1001 Genomes Resequencing Project, but these 200,000 SNPs were sufficient for GWAS. So we did 3 different approaches over 3 different studies. One was a multivariate genotype environment association, looking at correlations between multiple environmental variables and multiple genetic variables, multiple loci, that is. The next is a univariate approach where there's a single environmental variable and a single SNP at a time being tested. And then the last is an integrative approach where we synthesize evidence from genotype environment and multiple common gardens that were conducted across the range of Arabidopsis. From this, we take 90 candidate genes that were the top genes across these studies. We are interested in seeing whether we see any evidence that these top genes from these genotype environment associations are actually locally adapted. About half of these genes had available confirmed homozygous Salk tDNA insertions. And because we were interested in drought adaptation and we had been testing drought and moisture related climate variables, we conducted a drought screen in growth chambers. This is currently a preprint posted on bioRxiv. So I'm just going to highlight one of the candidates that came out from this. This is the very top SNP associated with the multivariate approach. You don't need to know exactly how this works in detail because it's a bit complicated, but we have a number of different climate variables and we have all of our 1,000 different ecotypes. And on the bottom right are the SNPs across the genome showing you how strongly correlated they are to either side of this climatic axis. We have precipitation seasonality characterizing a lot of that first axis, and our top SNP is highlighted. That SNP falls in Werke 38. This is a transcription factor, which in our drought screen showed an interesting phenotype of not accumulating anthocyanin under the drought. So perhaps being less drought stressed than the other plants, both wild type and mutant. When we compare the biomass responses and the fitness responses of these plants, we see that there's better drought response, better drought performance for the Werke 38 knockout. If you look, there's a big drop in biomass and fitness for wild type under drought, but for Werke 38, there's only a slight drop in biomass, and drought fitness response is pretty negligible, so that when we look in drought, actually the Werke 38 has higher fitness. This is work that's led by Yuxin Luo, my PhD student, with previous contributions from Claire Lortz, my tech. Yuxin has also done work with chlorophyll fluorescence and stomatal conductance to show some improved stomatal behavior in the Werke 38 mutant. We also see some interesting data in the 1001 Genomes Project resequencing, which is an extra dimension available now. We see that this SNP that we identified tags natural loss of function variants in Werke 38. That's the green alleles. Those are more common in the drier parts of Europe. You can see on the plot on the bottom left. The loss of function variants are more common where you have less moisture in the summertime. So that suggests that perhaps this tDNA insertion for Werke 38 mimicked the natural loss of function variants that are more common in dry environments that seem to be selected there based on their frequency. So are these approaches living up to their promise? Well, one of the limitations of a lot of current studies is that either they're just looking at environmental GWAS to generate hypotheses, but not testing those hypotheses, or people who are working on reverse genetics may be cloning genes and finding phenotypes, but perhaps not testing those in ecologically relevant conditions or under natural variation. So we're hoping to suggest integrative collaborative efforts where environmental GWAS generates hypotheses, but then these get tested with NILs, gene edited lines, or tDNA knockouts, for example. So I acknowledge co-authors and our funder, and sorry everybody for the slow start. Thanks for being here and teaching me something new about how to share screen.
Very amazing jobs. I mean, it's a very good work. Used the traditional genetic method to combine with the molecular work. Excellent. So please, Raji, welcome to our next moderator, Raji, and also Raji will introduce our last speaker.
Yeah. Hello everyone, I am Rajesh Sanyal. I'm doing PhD at National Institute of Plant Genome Research, and I've been a Plantae Fellow and currently working as a Plantae editor. And my PhD research work focuses on photosynthesis with a special focus on the plant development and the various transcription factors that might be involved in this process. So beyond the laboratory, I also love photography and of course networking. So now presenting our next speaker, Dr. Gaurav Zenta, who is a senior scientist at the CSIR at the Institute of the Himalayan Bioresource Technology in India. And he has been an assistant professor in the CSIR, where he is teaching several courses. And he has completed his PhD from the University of Antwerp in Belgium and postdoc at the Shanghai Center for the Plant Stress Biology in China. And he is a feature editor of Molecular Plant, associate editor for Plant Environment Interactions, BMC Plant Biology, and many other journals. The key area of his research interest includes the plant stress physiology, climate change, biology, the genome editing, epigenetics, and the chemical genetics. His lab is known as the Plant Adaptomics Lab, and I now want him to share more about his research. And yeah, the stage is over to you, Dr. Zenta.
Thank you, Rajshri, for this wonderful introduction. Let me share my slides first. Are my slides visible?
Yes, we can. Thank you, Dr. Zenta.
Okay. First of all, thank you, the organizers, for giving me this opportunity to present some of our work here in the Plantae webinar series. I feel, very privileged that I was also a Plantae Fellow in 2019, and now today as a faculty member, I'm here, presenting it in the webinar series. So the title which I have picked for my, talk is an integrative view on plant responses to climate change. So, so this is a quick, scientific journey. I did my PhD from University of Antwerp with two professors, Professor Ivan Janssens, who was basically an ecologist, and then Professor Hanazar, who is basically a molecular physiologist. Then I moved to Shanghai Center for Plant Stress Biology to do my postdoc with Professor Zhang Kangzhu. And then in May 2020, I returned back to India, and I'm here. I have established a lab here in India, an integrative plant adaptomics lab, which is in the biotechnology division at CSIR Institute of Biotechnology. A very brief glimpse of the CSIR, basically the organization where I work, basically. So we have a research organization. We call it as Council of Scientific and Industrial Research. We have 38 research laboratories across India. We have 39 outreach centers. And if you see about the leadership, the president of CSIR is basically our prime minister, and the director general is Dr. N.K. Kalai-Selvi, and our director is Dr. Sudesh Kumar Yadav. So if we see, we have basically a pan-India presence across India from north to south, from east to west. So basically, if we come to the mandate of my particular institute, that is CSIR Institute of Himalayan Bioresource Technology, it was established in 1983. We have basically a mandate of developing technologies for boosting bioeconomy through sustainable utilization of Himalayan bioresources. So in CSIR IHBT, we work across various crops, ranging from industrial crops, aromatic crops, medicinal plants, floriculture crops, and we have all the bioresources which thrive in the Himalayan ecosystem, from bacteria, fungi, algae, and we try to few products out of these bioresources so that we can contribute to the bioeconomy of the nation. So in our institute, we have basically 5 divisions from agrotechnology, biotechnology, chemical technology, dietetics and nutritional technology, and environmental technology. Let's come to the main agenda. Basically, the talk is around climate change adaptation and basically how plants respond and adapt to the climatic factors. Needless to say that plants are phototrophs, and all the life on Earth basically relies on plants because they are phototrophs. They fix carbon dioxide and gives us oxygen, food, and on the functioning of the plants, the whole ecosystem functioning happens basically. And they are basically sessile in nature, so they cannot move. For us as a plant biologist, they becomes really an important or excellent organisms to study stress adaptation because they are challenged with the various kind of abiotic stresses and biotic stresses in general. So needless to say again that climate change is real, and studying adaptation becomes more important when we have different scenarios of climate, CO2 emission scenarios from mid to middle to low. So all these CO2 emission scenarios depict that there will be rise in the global Earth temperatures. So the global surface temperature is increasing, and it is predicted that nights are getting more warmer, or they are getting warmer at the faster rate. And if you see the last couple of years, the Earth has really become hotter and drier. So climate change is impacting even the hardy plant species. So basically, this is one of the spotlight article which we published in Trends in Plant Science, giving an indication that plants thriving in the extreme Himalayan environment, which can thrive extreme cold, high radiations, are not able to basically cope up with the global warming now. So there are various levels of temperature rise, or different layers of temperature rise, or ranges of temperature rise. If you see, temperature rise can take place when we have a gradual rise in the temperature, or when we have the extremities in the temperature. So when there is a global average temperature rise, we call it as warming, and when the temperature is really high, it is called as heating or extreme heat. Plant perceive these temperatures very differently. Plant, when they are grown under warm temperature, it is perceived by phytochrome B, and this signaling cascade is mediated by PIF4, and a thermomorphogenic response is triggered. However, under high temperature stress, plants sense the temperature, result in the induction of the heat shock factors, and a thermotolerance is produced. So recently, people have shown that there is a crosstalk between thermomorphogenesis and thermotolerance pathways. So when plants are exposed to the stress episodes, they generate reactive oxygen species. So to cope with the the oxidative damage or the reactive oxygen species like hydrogen peroxide, singlet oxygen, superoxide, or hydroxyl radical, antioxidants are produced. So basically, when the load of the reactive oxygen species increases, it results into the damage of the lipids, proteins, and DNA basically. So in our lab, basically we work on a few crops. If you see this evolution of agriculture, in the ancient agriculture, crops like amaranth and quinoa and millets were very popular. Then it came an era of machine revolution. Crops like rice, sugar cane, tea came into existence, and then the green revolution came, where crops like wheat, rice, maize, and potato predominantly dominated this agriculture system. And today we have a time where we have gene revolution, where Arabidopsis thaliana, which is basically a model system, is really contributing to the growth of the to the growth of the knowledge, which can contribute to the enhancement of the crops. So in our lab, we basically work on the ancient crops like quinoa and amaranth. We also work on potato, which is basically a staple crop, and we also use Arabidopsis as a model system to dissect fundamental questions of plant biology. So let me give you a brief glimpse. I mean, I have, split my talks into different stories so that I can, at the end of the day, make you, can convince you that how, integrative approach can answer the fundamental questions of plant stress adaptation in the face of climate change. So this is the first story where we perform sort of meta-analysis to understand how elevated CO2 and stress factors interact with each other. So in this, study, we compare different species like C3 and C4, legumes, grasses, and trees when they were exposed to the, elevated CO2 in different growth facilities like grow chamber, greenhouse, open-top chambers, and phase systems. So the take-home message from this slide is that what we found basically, we found that elevated CO2, mitigates the impact of abiotic stresses in all these scenarios. So what are the basic mechanisms by which elevated CO2 protects from the stress factors? So we conducted an experiment where we challenged Arabidopsis thaliana. This is basically a story two, which I'm trying to give you a mechanistic insight, basically how elevated CO2 is protecting plants from the drought and heat episodes. So we exposed plants to carbon dioxide at the optimal conditions and elevated CO2 at 730 ppm, also to the combination of heat and drought HD, and through the combination of heat, drought, and CO2. And what we found here is really that elevated CO2 enhance the growth, and it impacts or it mitigates basically the impact of heat and drought. So when we did the global transcriptional analysis and also performed the photosynthetic analysis, what we found that indeed the effect which we find in the biomass, we also observed it in the photosynthesis and the chlorophyll fluorescence. And the ROS production was increased in the stress conditions, but it was basically at the lower level in the elevated CO2. So when we looked into the photorespiratory processes, basically photorespiration happens in three organelles, in chloroplast, peroxisomes, and mitochondria. So, and it is basically a site of the hydrogen peroxide production, which is basically a ROS. So we quantified photorespiration indirectly by using biochemical metabolites like glycine and serine ratio. What we could infer from this is that under elevated CO2, there is less photorespiration happening, so which possibly result in the less ROS production during the stress episode. This is how it can contribute to the stress mitigation effect of elevated CO2. So the next story, which I'm trying to tell you, is on Chinopodium quinoa, basically. It is basically a super food. I mean, nutritionally very rich, and at the same time, it is very resilient to various stress factors like drought and salinity. However, people have demonstrated that quinoa is highly heat sensitive, but people have not dissected the mechanisms why it is really heat sensitive. So we set up an experiment where we challenged Chinopodium quinoa to day heating of 38 degrees and night heating of 32 degrees. And what we interestingly found is that night heating is indeed very detrimental for the yield of the quinoa seeds. So when we looked into the transcriptional and microscopic analysis, and we also complemented these data set with our physiological data, what we could see that the gene ontology, the geoenrichment says that photosynthesis, cellular respiration, and redox homeostasis were predominantly coming up in these geocategories. So when we looked into the photorespiratory pathway, we could find that daytime and nighttime triggers asymmetric effects on CO2 assimilation and evolution pathways in quinoa, which basically results in the more carbon losses during the night temperatures. So the next story which I am bringing forward to you is on potato, basically. Why potato is important? I think it's an excellent model system to study source and dynamics. Basically, carbon is photosynthesized in the source tissue in the leaves, and this carbon is basically dumped into the stolons, or you can say the tubers. So various environmental factors like short day, long day, or heat has a control on the tuberization process. So our lab is primarily interested in understanding the impact of high temperature in tuberization. And heat basically is a negative regulator of tuberization. The moment heat increases or the temperature increases, is basically impede the process of tuberization. So basically, this is the regulatory network which demonstrate the complexity of tuberization. And it also demonstrates that under high temperature stress, less of the molecular regulators has been identified and has been validated. So in our lab, we try to compare the sensitive and the tolerant genotypes of potato, and we could figure out that the sensitive genotypes are unable to tuberize under the high temperature regime, whereas the tolerant genotype is able to produce tubers. So we did some sort of Ross accumulation assays by staining through DAB and NBT in the leaves, and we found that the tolerant genotype, the accumulation of the raw species is less. So we performed the transcriptional analysis, and we found that the ascorbate peroxidase 2 is predominantly overexpressing in the tolerant genotypes. We did a lot of molecular biology assays like localization, basically where it is localized. We found that it is localized in the cytosol. We did some heterologous experiments overexpressing these genes in Arabidopsis, and then quantifying how they are basically responding to heat stress. To cut the long story short, what we found that when we were overexpressing APX2 in Arabidopsis, we found increase in the thermotolerance in terms of survival and less accumulation of the ROS. And the next story, which I'm touching upon, is the effect of transgenerational heat stress adaptation in Arabidopsis. So Arabidopsis is really an important model system in this context, because if we want to understand how long-term stress exposure impact basically the fitness of the plants. So because of the short generation time of the Arabidopsis, we can test these hypotheses in multiple generations. So in this experiment, we did the transgenerational heat stress experiment, where we exposed Arabidopsis to the for 4 generations to 38 degrees, and also we kept few plants in controlled conditions for multiple generations. At the end of the experiment of 4 generation, we performed reciprocal transplantation experiment, where we put the plants which were coming from the heat selection to control stress, and the plants coming from the control selection to the heat stress. So interestingly, what we found is that heat selected genotypes evolved as more thermotolerant genotypes. When we performed the gene expression analysis, we found that the heat selected genotype had the constitutive expression of the basically defense genes. And when we perform the bisulfite sequencing at the whole genome scale, we could link these expression patterns with the methylations. So giving us a hint that epigenetics plays a crucial role in long-term stress adaptation against the heat stress. So in the another story, we tried to demonstrate it in a more natural population of Arabidopsis, where we compared Arabidopsis accessions col0 and C24, and we performed whole genome bisulfite sequencing in these accessions, and also in the crossings, reciprocal crossings of these accessions. And we could find one DMR region which basically was differentially methylated in these two accessions. We did the fine mapping, and we could precisely localize where is this region, and we identified this region as NMR19. And by using diversity analysis, we could formulate a sort of population structure for this particular DMR. And at the end of the day, we could demonstrate that this is an epi-allele which is stably inherited from one generation to another, and it is linked with the temperature of origin of the accessions, and it is basically controlling temperature-mediated leaf senescence in Arabidopsis. So the overall conclusion which I want to draw from my presentation is that elevated CO2 mitigates the impact of climate extremes, and day and night temperature heating have asymmetric effects on elevated CO2 assimilation and evolution pathways. Redox regulations play a crucial role in stress adaptation. Epigenetic modifications such as DNA methylations are key mechanism in plant adaptation to climate change. I would like to thank my lab members, the postdoc fellows, and the PhDs and the trainees who has contributed to this, this data set which I have just shown to you, and all my funding from CSIR, SERB, DBT, IIT Mandi, and HCI Foundation, all the collaborators and director CSRI HBT Palanpur. And thank you very much for your attention.
Thank you, Dr. Zenta, for this wonderful presentation. Let me share the slide. Yeah, now we come to the question and answer session. So I request all the audience to ask their questions in the question and answer session box, and we will take the questions to the moderators. And I request Dr. Howe to convene the session.
Hello, everyone. Firstly, I acknowledge the answers from all the speakers. I mean, the speaker have already answers a lot of the questions. And firstly, I will fast share the response of this question quickly, and then we ask the more questions. And there is the one question for the Dr. Justin about the people think maybe it's for the conifers, the Christmas trees, they are not most consumed by the humans. And how can Dr. Justin convince the policymaker or the citizens that the conifers are as important as the other plant? And Dr. Justin gives the very clear answers to provide the usage and the applications of the Christmas tree and how Christmas tree are managed and cultivated, and also emphasize the economic importance of the Christmas trees. And you can check the QA panels to see the details, the explanation of the doctor Justin. And also the Dr. Lasky give the answers for two questions. And one question is about the environment may modify the gene expression due to the epigenetic. And is that true? And Dr. Lasky give the positive answers. And the second question is some genes should be low because they are switched on off according to the environment, and also it should be low for the structural properties. And Dr. Lasky also gave the positive answer. And besides these questions, we will give the other questions to all of the speaker. Firstly, the first question will to the Dr. Li Yan, our first speaker. And the question is, how to shift in the phosphate allocation, particularly the consistent metabolite phosphate, and reduce the lipid nucleic acid phosphate benefit the plants with the delayed leaf greenings in the phosphate limited environment. And yeah, Dr. Yan, can you hear the question?
Yes, I just hear the question, but I can't see it. It's a very good question, and one of my studies we noticed the delay greening. I would like to introduce that briefly to everyone. You may notice on the street or some of the trees or shrubs, they have very bright young leaves on the top, like red or yellow or purple. So this is not a poison or like a nutrients. Normally these species, they are very smart to have a high phosphorus concentration when they are very young, but during the leaf development, the leaf phosphorus concentration will reduced and reallocate to other cell structures. So we do notice that, and we found for many species, the young leaves, they have a very high concentration for nucleic acid, and some species they have very high phosphorus for lipids. So this will be very important for them for, as before the leaf will develop, they can't very well protect themselves for the strong sunlight, and not very efficient for photosynthesis. That's why they have this kind of a strategy to allocate phosphorus differently to different fractions. But we only did one study, and in 6 species, I think we will need a larger scale for more species and more data about that. And to answer another question is, I think some plants they are very smart and like proteaceae, and they can hold a leaf totality like 0.3 milligram per gram. That's very, very low compared to other locations like China or America, as I know. But they are very efficient to function well with photosynthesis with this kind of very low leaf phosphorus. And one of the strategy is that they can efficiently allocate more phosphorus, not to phosphate, not to PI, but to metabolites or nucleic. As you will need lots of proteins to maintain and enzymes to maintain your function for photosynthesis and respiration. So that's why we think it's very important to figure out how this kind of large scale, or it's commonly for many other plants like pines or trees or even invasive plants, can they do the similar things? So yeah, thank you for the question.
Yeah, and the second question is to the doctors, the Whitehills, and this question is how it can be managed to merge the public interest in having the softer plants in their home with the high resistance to the pathogens. The question, the audience assumes the physical hardness of the conifer leaves can help them to against the pest and the pathogen. So, I mean, the audience just thinks that there might be the contradictory roles of the hardness of the leaf in the conifers to the pathogens and the pest. And Dr. Whitehill is trying to type in the answer. Maybe the Dr. Whitehill can answer it lively here.
Yeah. Yeah, I was just typing my response, but I'll just read it. I was saying that the physical hardness of the needles don't really appear to be an important trait for protection of these trees against any pests or pathogens. Fraser fir is kind of unique amongst most conifers, in that the only major insect pests that the species is threatened by are piercing sucking insects, and these insects use a much more stealthy way of attacking the plants. Instead of like bark beetles that chew on the plants, these piercing sucking insects, there's four to 10 different species that we're dealing with in North Carolina, but they use a stylet, long straw-like stylet, to enter through stomates on the underside of needles and weave in between cells to get to an individual cell that they feed on. So the insect pests aren't really going to be influenced by that. And Phytophthora disease primarily swims through water and is attracted to the sugars and peptides, proteins, root exudates that are found in the soil, and they are able to sort of hone in on the plant and then cause infection through the root system. They can maybe attack aerial parts of the plant, but it's very uncommon. And we definitely don't see anything through the needles. It's usually primarily through the roots. And then the last one, the deer, which are a big one, is they feed on trees during the cold winter months when there's no food left, and they don't feed on the needles. They actually feed on the very kind of nutrient dense buds, which would then grow into branches in the following season. And so you may have a 4-year-old tree that's 3 or 4 feet tall and covered with hundreds of these little buds, but the deer will go through a plantation and chew all those off, and that's how they get their energy, amino acids, proteins during cold winter months when there's no other food and all other food sources are sort of scarce. So yeah, it's a great question, but the kind of the biology and the ecology of all these are sort of difficult to, yeah, nothing that we're dealing with would probably benefit from that at the moment. So
Okay, great answer. And the third question is for our third speaker, Dr. Laskis. And as we know, there is several drought adaptation strategies, such as in Arabidopsis, such as the escape, avoidance, and tolerance. I just wonder, could your GEA approach effectively distinguish the gene loci associated with the different drought adaptation strategies in Arabidopsis? Dr. Lasky?
Sure, that's a good question. I don't know that it would be very good for that to distinguish between the two. It's kind of hard. I think that if we knew something about the phenotypic effect of a given mutation, then you might be able to tie that potential phenotypic effect to one of the strategies, but that's often unknown. You might assume, based on knowledge that you've already gained, that Arabidopsis or whatever species you work on, the populations in certain climates tend to adapt via certain strategies. Let's say a certain climate seems to favor escape strategies. So then you could perhaps use that information to then say, well, I'm going to assume that the alleles that are associated with those environments are conferring those strategies. But, there's a bit of speculation there that you, of course, would want to test with experimental follow-up.
Great answer. Yeah. And we have the last questions for the Dr. Zantar. And the question is, can plant be smart enough to tackle the abrupt change in the environment?
I think it's a wonderful question. Yes, I mean, there are multiple, papers demonstrating that, plant can react and respond to the environment very quickly. There are, papers from Ron Mittler lab where they have done, one paper is in New Phytologist, I guess, and another one is in Plant Journal. They have demonstrated ultra fast responses to the heat stress, basically, and jasmonic acid. So in the second speed, basically plants can respond and can equalize with the changing environment. So definitely, I mean, plants have evolved in such a way, they can quickly adapt to the environmental perturbations.
Great. And our moderators may also have some questions. And if you want, I mean, so you can ask directly, the Yuan or Raji. Do you willing to ask some question here?
Thank you, Hao. I just have a quick question for Dr. Lasky. Besides convolutional natural network, what other deep learning network can be used for the prediction of the de novo cis regulatory motif in DNA sequence that are important for gene expression?
Yeah, I don't know. That is worth pursuing. I'm not familiar enough with the wide range or potentially new approaches that are out there, but it's certainly something worth pursuing.
Okay, thank you.
And Raji, do you have any question? You are muted. Raji, you are muted.
Sorry, yeah. So all the questions have been properly answered in the question and answer session, as well as the panelists are constantly writing to all the questions from the audiences. And as I can see, we are running a bit late. We are running out of time, and so it's better to conclude the webinar because people, the shorter it is, it will be more impactful. And from all the studies, from all the speakers we heard, we started with Dr. Yan, who talked about phosphorus utilization and their allocation and how it plays an important role in the species distribution. And then Dr. Whitel talked about the Christmas tree species preserver and how they are doing the genetic improvements for better resistance and for making it like available to the people for decorative as well as the for the economic impact of it is also increasing the economic impact as well. And Dr. Lesky then talked about an novel method, innovative method, which is the genotype and the environment association, which is similar to GWAS with the environment. And it's a good way to find the locally adapted genes. And they've also found a good gene, WORK38. So you can obviously read all the papers from these authors and have more insights on their research. And finally, Dr. Zinta talked about the various aspects of the plant in different crop species like potato. He also talked about the model plant and the role of the elevated CO2, the ROS production, as well as the translational heat stress. And so this can be very innovative methods which all the people can apply to their research for identifying the important genes which are effective in this response to the climate change. So yeah, finally, I want to thank all the authors for their valuable time, and I want to hand over to Jason for concluding the webinar.
All right. Yes. Thank you again to our speakers. Such great content. And thank you to our moderators who organized this webinar. And of course, thank you to all our audience for being engaged and asking really good questions. Again, this webinar is recorded and you'll be able to watch it later on once everything's processed and posted to the webinar blog post. Other than that, I hope you all have a great week and we hope to see you on the next section. Have a great day, everyone.
Thank you.
Bye.
Okay,
Thank you.
bye-bye. Thank you very much, everyone. Bye-bye.
Thank you,
Have a great
everyone.
day.
You.
Thanks. Bye.